familiar with Federico's work for a very long time. When I was a grad student, I was reading all these papers coming out of the Gordon Lab, including papers by Federico, and so they definitely helped shape me into the microbiome scientist that I am. If you are not familiar with Federico's work, it's really excellent, comprehensive work using autobiotic systems to interrogate how the microbiome and host are related. He uses all sorts of different techniques, not just 16S and metagenomics, but lipidomics, metabolism, all of that to get really comprehensive pictures of what's going on in these autobiotic systems. One element of his work that particularly strikes me where I'm interested in is his use of genetics as a tool to kind of interrogate these interactions. So in these mice systems, he's able to use the host genetics of the mouse to kind of figure out what is going on between some of these metabolites and the bugs in the guts of these guys. So I'm really thrilled that he's able to join us today. And as a token of our appreciation, we give all of our invited speakers one of these little plaques. It is personalized. This is an example, but yours will have your name on it. I'll turn it out to you.
Thanks to Abby for the personalized card.
Yes.
Thank you, Abby.
Sitting around here, yeah.
Thanks so much for the invitation. Nice introduction. It's a pleasure to be here this morning, sharing the sort of work from my lab. And so this is a nice. This one I'm from. So my lab is in the University of Wisconsin-Madison, and so our lab is from here. And we're in front of these two nice, sort of like surrounded by these nice lakes, and now ice cubes. A lot of ice fishing going on right now. That's sort of I'd like to tell you sort of about like sort of the work that we're doing, and so we're just going to cover sort of pretty much sort of the extremes of what we do in the lab. So we're talking about causes and consequences of gut microbiome variation. So I don't think I have to sort of do much for an introduction of microbiomes here, and just sort of like sort of say that it's, when we look at the microbiomes from the gut of different individuals, we see sort of this large variation, and sort of we are interested in this variation. So what causes this variation, and sort of what are the consequences of it? And so we know that environments or particular diets and sort of genetics sort of contribute to this variation. And today I sort of like to basically tell you two stories. One where we take advantage of sort of system genetic approaches to learn about connection between microbes and the host, or microbes, metabolites in the host. And the other one is sort of like it relates more to our work on cardiovascular disease and understanding how sort of differences in microbiome composition affect predisposition to cardiovascular disease. And so I think sort of I don't need to do a lot of convincing here, to get the idea that host microbe interactions are complex. They have many levels of complexity. And these sort of include, for example, the great diversity of organisms that can colonize the gut, each one with unique metabolic sort of potential. So for example, we look at, this is sort of the metabolic network of E. coli. Think about, and these are the organisms that we know the most about it. There are 200 different species at least that colonize us, each one with metabolic networks at least as complex as this one. So sort of that level of complexity combined with sort of the interactions that happen between different microbes in the gut, with the host, with the diet. And so sort of, we're trying to understand, sort of think about how can we leverage, how can we break down this complexity. And so, we know sort of this evidence now, so several lines of evidence suggesting that host genetics modulates the composition of the gut microbiome. So, the idea is sort of to take advantage of these sort of genetics and use genetics as an anchor to start the computing these interactions. And so, we're using a mouse system, so sort of genetically diverse mice in which we collect, many different traits. So these involve, some of them are microbial traits, taxa, microbial taxa, or sort of microbial genes through metagenomics. We collect information about metabolites, gene expression of, it can be gene expression of the host. And then for each one of these mice, we have genotype data, by using high density SNP arrays. And so then what we're doing is basically do quantitative trait locus mapping. So basically, we're trying to identify regions of the genomes that contribute to the variation that we observe in these, phenotypes that we're measuring. But the idea is try to identify these loci that contribute to the variation in, in traits, so for example, microbial traits or metabolites. But what we're going after is these traits that control, loci that control both microbial and metabolite traits. So, the idea is when you find a locus that control traits that are not previously known to be related, you can come up with hypotheses about how these traits are related, and design experiments to test these associations. And so, giving you more details about the mouse cohort that we're using is the Diversity Outbred population. So this is a heterogeneous stock that was generated by Jackson Labs about a bit more than a decade ago from 8 inbred strains. So there's these 5 common lab strains plus 3 wild derived strain, and so they came up with this outbreeding strategy to generate thousands of mice, where each mouse is unique and their genome is basically a mosaic of the, A founder strains. So these mice were bred to maximize allelic variation, and combined this cohort has about 40 million SNPs, which is comparable to what human population has. And this is what I was telling you, that when we look at each DO mouse, this is how they look, where each genome is unique mosaic of the A founders genotypes, and we can map what allele, what the founder alleles are present in each locus. Okay, so then we got. So here I have 300 mice, but data that I'm going to show you today is between, depending on the assays, between 300 and 500 animals that we use for the metagenomics. Specifically, we use 300. And so then these mice were purchased in batches of 100 from Jackson Labs, housed individually for 22 weeks. There are a number of other phenotypes that were measured, and this I want to point out that this is a collaboration with actually Jackson Labs. Alan Addy, who's been the driver of this in Wisconsin, that he's interested in metabolic disease, and he's hunting for genes involved in type 2 diabetes. And then Carl Broman, who basically wrote the book on QTL mapping, and Josh Kuhn, that helped us with the metabolomic studies. And so there are large data sets in this. I'm just going to focus on on a few of these that we've been leading. So the idea is we take these DO mice, we collect feces for where we do shotgun sequencing, and so when then we do de novo assembly, and so we can basically map either metagenes from these organisms. We can map abundance of genomes, and here, so these are the traits that we're mapping. One, we can map all these metagenes. We can beam genes into KOs and map the abundance of the KOs, and we can also map, as I said, the abundance of the genomes that we assemble from these metagenomic data. So what I'm showing you here is data mapping where different KOs. So this is like a Manhattan plot, and each dot represents a different KO, and the position is where they map, where their abundance map in the the mouse genome. And so above, these are the ones that are significant after, fold discovery, sort of rate and so on. And you can see that we have some hotspots, for example, K there. So it's, all the KOs that map there are all genes involved in sporulation. So we are pretty confident that that's the, that region of of the genome controls abundant sporulation genes. We can also sort of map taxa, and and these are different, each dot in this case is a different taxa, and so below 8 sort of like are all significant associations with the mouse gene. But what I told you is that we want to use sort of genetics as an anchor to connect phenotypes. So one of the phenotypes that we're looking at, one of the traits that we're looking at, are these sort of genomic information. The other one that we look at was lipidomics. So we basically collected so this is from 400 mice. We collected so untargeted lipidomic data. So using mass spectrometry. So you can measure about thousands of different lipids, spanning 25 different lipid classes, and a lot of these sort of lipids are dark matter, things that we don't know what they are. I mean, we can annotate a very small fraction of these lipids. And so this is, again, so this is, what I'm showing you here is sort of the diversity in terms of, retention time, sort of my HPLC, MS, and the diversity sort of in mass, among the lipids that we're measuring. These are, this is a classification of everything that, we are quantifying. And, I want to sort of note that, sort of the largest class, group is sort of things that we cannot annotate. We don't know what they are. The other thing that I want to sort of point out is that, we see huge variation in the abundance of these lipids across the, 400 DO mice. This is sort of just looking at 3 different lipids across sort of the 400 mice. You see that, some mice, that don't have almost anything of these lipids, mice that have a ton of these lipids. And so, expanding sort of several logs, in terms of the signal we get. And so we can do the same sort of QTL analysis, and, we can find that, where each dot here represents a different lipid species, and we can find associations between, lipids and the mouse gene. But sort of what we're trying to do is now connect these two sort of data sets, again, using genetics as an anchor. So one way that we started to do that is sort of with one mediation analysis. So I think sort of mediation analysis is like, testing the idea whether, the presence of a lipid sort of mediates sort of the mapping of an organism to sort of specific logs. And so the way we do this is we run our QTL analysis using, the potential mediators as a covariate, and compare sort of the results with the initial QTL analysis. So if there's mediation, you expect to see a drop in the mapping signal, a drop in the significance. So we basically tested all the lipids as potential mediators of the mapping of the bacterial gene. And so found something like this. So this is the sort of a score for certain set of genes. And then when we use this lipid as a potential mediator, we see sort of a change in a big drop in the sort of significance of a bunch of genes. And actually, sort of we have 3 lipids that cause a shift in the mapping of sort of genes that belong to Akkermansia muciniphila. All these sort of dots here are single genes of Akkermansia muciniphila that their mapping changes when we use these lipids as the covariate to do the QTL analysis. I mean, we can also sort of show this way, sort of saying that when we look at the Akkermansia muciniphila genome, so here's what it maps. We see that the cecal lipids map in the same place. So these lipids mediate the mapping of Akkermansia, and they co-map to the same locus. But so this is usually not enough information because these data tend to be noisy. So this is the idea. So like we have this hypothesis that these Akkermansia and these lipids were somehow related. That's what we went on to test. And the reason we thought, and I want to sort of point out that we found several examples like this. We focus on these specific ones because of the organism involved in this sort of relationship, sort of Akkermansia muciniphila. So this is an organism that is commonly found in the intestine of mammals. It's famous for influencing sort of host metabolism. It has been negatively associated with metabolic disease. So it's, we think of this as a very beneficial, pretty beneficial organism. There are probiotics out there that you can buy. There are people making money of this bacterium already. And actually, there have been clinical trials where they show that this organism lowers GLP-1 and so increases our GLP-1. So it has sort of many beneficial effects. This organism has also the interesting feature that lives using mucins, so that produced by epithelial cells. So it lives in close proximity to the gut epithelium. Other human studies have also found genetic association and sort of has anti-inflammatory properties. Okay, so this sort of organism come up with this unknown lipid field. So we did some further analysis with mass spectrometry and figured out that these lipids were ornithine lipids. And so I'm going to tell you about a little bit about ornithine lipids. So they look like this. So you have your fatty acid chains, but instead of having a phosphate group and having phospholipid, they have an ornithine head. So they tend to be less polar than phospholipids. These are phosphorus-free lipids that tend to be present in the outer membranes of gram-negative bacteria. And actually, sort of the way that this is, that most bacteria make these lipids is, or not most bacteria, that the bacteria that makes this lipid, produce them, is usually under phosphate-starving conditions. There's no phosphate, they make these lipids. There are, set of bacteria that can make these lipids. And then another sort of interesting feature that they have is that they have these acyl-oxy-acyl structures right there that actually is similar to lipid A in LPS, which is sort of a structure that is recognized by toll-like receptors and triggers sort of an innate immune response in some mammals. And so here's LPS for you on acyl-oxy-acyl. And sorry, I don't see very well. So if I'm pointing anywhere, it's because I don't see, my eyes are not very good, and I don't see the pointer. Anyways, and so there's some work, ornithine lipids have been seen before in other organisms, not gut bacteria, but for example, there's a study from the 80s that, showed that ornithine lipids interfere with LPS-induced upregulation of inos and COX-2 macrophages. And there's another study, crazy study, where they took these ornithine lipids and gave it to mice before they gave LPS, and they found that it prevents the lethal effects of LPS. So this sort of, I think there are probably 2 or 3 organisms known to have these ornithine lipids. So, for example, agrobacteria make these lipids under phosphate limiting conditions. And, for example, this paper showing that agrobacteria that do not make ornithine lipids induce rapid tumor formation. So there's some precedent for these lipids to be sort of important for microbe-host interactions. Okay, so we see these sort of lipids now come up in Akkermansia. We see these lipids mediating the genetic mapping effect of Akkermansia. So the obvious question is whether these these 2 traits are related. That's the obvious question was, does Akkermansia make these lipids? And so that's exactly what we tested. So here in terms of mass spectrometry data for one of the ornithine lipid types that we see in the cecal of the mice. And this is our blank signal, this is B theta and E. coli, 2 other gram negatives, and this is what Akkermansia muciniphila makes. So this is lipid extracts of in vitro cultures. We can also colonize mice, germ-free mice with Akkermansia or other bacteria, and we see that these lipids are absent in germ-free mice and only present in mice colonized with Akkermansia muciniphila. So the other thing that I told you is that these lipids are made by bacteria when there's no phosphate around, right? So we tested that idea in Akkermansia mucinifila, and actually that is not the case. So this is the list of all the ornithine lipids we detect across different phosphate concentrations. We see, there's a little bit of a change, but for the most part, Akkermansia mucinifila makes these lipids no matter how much phosphate there's around. So 1 millimolar phosphate is a lot. So this is sort of like 0.02 millimolar is a condition that basically limits growth, and it's not enough phosphate even to double. Anyways. So this seems to be central to the biology of Akkermansia. So everything that I show you so far is in in mice. So we look at humans, samples, fecal samples from humans that for which we have 16S data, and we have sort of Akkermansia abundance data. And so we measure abundance of the ornithine lipids, and we see that the abundance of the ornithine lipids correlates with the abundance of Akkermansia in the samples. And so we think that at this point, we think that Akkermansia is probably one of the few, if not the only bacterium in the gut that make these lipids. So I think the reason I thought that this was interesting is because I mentioned the beneficial effects of Akkermansia mucinifila, and then this idea that Akkermansia makes a living consuming host-secreted glycans, so these mucins, and lives in close association with the host. Question, why doesn't this organism cause trouble or get killed by antimicrobial peptides? So it's something that I would love to do is test, make mutants that cannot produce these lipids in Akkermansia, but this is not something that we can do at this point. But there was this interesting study in Pseudomonas aeruginosa, actually. So Pseudomonas aeruginosa actually makes these lipids under phosphate limiting conditions. And this study, basically, they trick Pseudomonas aeruginosa to make these lipids under all conditions. And what they found is that, when they made Pseudomonas made these lipids, now the cells become more hydrophobic, and they reduce the bacterial susceptibility to antibiotics and antimicrobial peptides. And instead of being a pathogen to a host, it became less virulent and reduced the production of inflammatory factors. So it seems to be important for here's our hypothesis. We think that these ornithine lipids are important for the lifestyle of Akkermansia and interactions with the host. So we cannot do genetics. What we can do is synthesize the lipids. And so that's what we did. So, and as I told you this, we synthesized this lipid. And as I told you, it has these acyl groups that looks like LPS. And then we tested whether it impacted production of cytokines from LPS stimulated bone marrow derived macrophages. So we took macrophages from bone marrow derived macrophages or we generated bone marrow derived macrophages from mice, from B6 mice. And then we stimulated with LPS or LPS plus different concentrations of ornithine lipids. And when we see for example, this is production of TNF-alpha, we see when we add the lipids, they bring down production. This is LPS brings up production of TNF-alpha. So when we increase the concentration of LPS, TNF-alpha goes down. Similar situation we see for interleukin-6, when these ornithine lipids reduce production of these pro-inflammatory cytokines. So we also tested this. So I tell you that the way we found this was through genetic mapping, right? And so, and I told you early on that we can find what founder alleles, one of the 8 founder alleles, is driving the signal, the genetic signal that we're seeing. And so we found that 129 and the black 6 alleles were driving the mapping in different directions. So we generated bone marrow derived macrophages from both B6 and 129 mice, and we did the same experiment that I just described. And, we see some, some common patterns where, for example, in both cases, the ornithine lipids reduce production of interleukin-6, both in B6 and 129. In both cases, the lipids increase production of interleukin-10. But then, for example, when we look at interleukin-12, it's a completely different situation where, sort of there's not much stimulation of LPS, it's sort of interleukin-12 by LPS, whereas we see this sort of completely different pattern, on interleukin-12 in 129 months. So, it seems that, we see an effect of the effect of ornithine lipid on cytokine production sort of differ between B6 and 129 mice. And these differences in Akkermansia between the different strains might be related to, sort of how these two strains are responding to the lipid. But this is sort of like what we're trying to figure out. So in order to get, to understand how, sort of like get to how the host is responding to, Akkermansia and the lipid, we also did eQTL analysis. So basically we profile in the same DO mice that I described, the transcriptome of this small intestine and map the expression sort of different genes. And what we found was sort of this co-mapping of this gene called HTF3 with the ornithine lipids and with Akkermansia. So, HTF3 is a very important sort of transcription factor, sort of that sort of key in innate immunity. It protects against LPS induced inflammation in mice, and it negatively regulates production of interleukin-6 and interleukin-12. And so we're seeing that HTF3 is now co-mapping with the ornithine lipids and Akkermansia. So what we did was we took these bone marrow derived macrophages and we profile expression of HTF3, and we see that sort of the ornithine lipids increase the expression of HTF3. So in both cases. So now what I'm sort of going to show here is the genes. So these are the genes that HTF3 regulates. This is not our data. This is a release of genes that are known to be, regulated by HTF3. And so now we look at how the ornithine lipids control the expression of these genes in B6 and 129 mice. And what we find is that basically completely different patterns. So, here it is B6, here is 129, without or with the ornithine lipids. See that, the ornithine lipids sort of cause completely different signals depending we obtain the macrophages from B6 or 129 mice. So suggesting that sort of the lipids are signaling through HTF3, sort of the same way in both genotypes, but then the transcription sort of program that HTF3 unfolds is different for the two different strains. And that probably is what causing different levels of colonization of Akkermansia. That's what's kind of our hypothesis this time. So we see sort of Akkermansia muciniphila and ornithine lipids sort of co-mapping with HTF3, so regulating HTF3 expression, and then HTF3 regulating host immunity in ways that vary, as a function of host genetics. That's sort of what, sort of where we are, and sort of what, we're interested in finding more about, these sort of communications sort of have. So what I told you sort of so far is sort of bacterial taxa and functions, or told you about these bacterial taxa and functions associated with host genetics. I told you that Akkermansia muciniphila produces these ornithine lipids that are immunoactive. And so this is the first example of gut bacteria making ornithine lipids. And we found this, that the abundance of these two traits, the lipids and Akkermansia come up with the expression of immunity genes. So I show you only HTF3, but actually there are 2 or 3 other genes that come up with Akkermansia. And so we think that these ornithine lipids could be an important factor for lifestyle of Akkermansia and sort of in the gut and sort of interaction with the host. The fact that maybe it makes Akkermansia more hydrophobic, and more resistant to antimicrobial peptides may be important for the lifestyle of Akkermansia in sort of in close association with the mucosa. And so I think sort of the other sort of the thing about this is the open sort of the idea of how to use system genetics to establish sort of to draw novel connections between different traits. And actually, this is sort of an approach that we're starting to use. We are, I just sort of show you one. We have dozens of these connections that they are a little bit more complex than this one, to at least sort of initially the company. But there's a lot of sort of new connections that we're making. So I think so that is the first part of my talk. So and now I'm going to move into the understanding sort of the work that we're doing towards understanding how variation in the gut microbiome contribute to the different phenotypes that we are observing. And phenotype that my lab is really interested in is cardiovascular disease, specifically atherosclerosis. And the work I'm going to describe was mostly done by now Professor Kazuki Kasahara. So he started his lab last summer, and a senior scientist in my lab, Bob Kerr. And so, just sort of like to bring everybody to the same place, sort of, atherosclerosis is sort of a chronic inflammatory disease that is happening in all of us, which basically sort of entails the accumulation of fat and inflammatory cells in our blood vessels, more prominently in larger blood vessels like the aorta or the carotid arteries. And sort of these sort of plaques that we call them, sort of tend to increase in size, and they're full of inflammatory cells and lipids. And they can, if they grow too big, they can occlude they can occlude the circulation. They make our heart work harder, and so causes or they can break. And so when they break, sort of they can clog a smaller artery somewhere, and so you have a stroke, for example. So this is a chronic, progressive disease. It's a primary cause of heart disease and stroke. And, sort of like in the field has mostly think about this disease, cardiovascular field, as a disease of high cholesterol and sort of as a primary driver. But we know sort of environments and genetics play major roles. So we get sort of interested about this several years ago, and sort of we started collaborating with our Jake Lucius. Sort of he's at University of California, Los Angeles, and then sort of what I'm showing you, and so one thing that I want to mention is the way we assess progression of cardiovascular disease in the mice is by measuring the size of these lesions. So a small lesion, healthy mice, big lesion, we're in trouble. So Jake Lucius had been hunting genes for atherosclerosis for more than 30 years. And what I'm showing you here is sort of data on the size of the plaques for 100 different strains of mice that were all in the same diet. So these mice are all eating the same, yet some develop these huge lesions, some they are super healthy. And so Jake has been saying, for 30 years, the genetics is it. So then, I was lucky to meet Jake through a collaborator, and we get in this argument that, genetic was nothing, and sort of like, and I was partly kidding, but then say, I'm going to prove you that. So give me some samples, collect me some samples of fecal, and so actually we collected fecal samples from these strains, and first show that, the differences between these strains was not just genetics. The microbiomes of these mice. So here I'm showing that, the phylum level, the microbiome for the 100 strains. There's quite a bit of variation, among the 100 different strains. So we hypothesized that sort of this variation contributed to the, phenotypes that, sort of he was seeing in his mice. So I convinced him to send me some samples from these mice. So, this is what, sort of what we were able to get. Some mice that have some large lesions, we're going to call them atheroprone, and some mice that were, we're going to call these guys atheroresistant. So they develop like relatively small lesions. So then we took this, microbiomes. We use germ-free mice. And so we use, for these studies, we use germ-free ApoE mice. So these mice, I mean, they're used a lot for cardiovascular studies. They cannot handle cholesterol well. They have, levels of cholesterol that causes them to develop these plaques in about 2 or 3 months, as opposed to 30 years that, it takes in humans. So, otherwise mice sort of like have hard time developing cholesterol. So, these mutations are important. So then, we took these germ-free mice and put the microbiomes from the atheroprone and the atheroresistant and sort of keep them in the diet sort of here for 12 weeks. And at the end of sort of the study, we look at the lesions. So we found that mice colonized with the atheroprone strains develop larger lesions than those mice colonized with the atheroresistant strains. And so I also told you that atherosclerosis is an inflammatory, sort of chronic inflammatory disease with accumulation of inflammatory cells inside the plaques. So sort of like here are stained in brown, so for monocytes and macrophages. So we see that also mice that receive the microbiome from the atheroprone strains have more higher accumulation of monocytes and macrophages compared to the ones colonized with the microbiomes with the atheroresistance. So that was sort of a kind of foundational work for our lab, and so and sort of motivated for the study. But so the obvious question here was like, sort of how that is this happening? And there were some studies suggesting that certain microbes and certain metabolites associated with cardiovascular disease. We've tested all of those, and we didn't find anything that actually was convincing or anything that explained the differences that we're seeing between the strains. So then we took more of an untargeted approach, and so we did untargeted metabolomics in the plasma of these mice. And what we found was sort of like correlations we measured about 700 metabolites, and we found sort of a correlation about 18 metabolites that were positively correlated with plaque size. And many of these metabolites, as you can see here, are involved in sort of purine metabolism. And so to us, that was sort of kind of interesting, to see multiple metabolites in the same pathway possibly associated with disease. And the fact sort of that, that uric acid was there, that was also interesting. And so this is just showing the sort of correlation plots. Because, so the reason sort of that, uric acid was interesting to us is because hyperuricemia has been previously associated with cardiovascular disease. And so you might have heard about uric acid before. That's sort of like uric acid, sort of is, the waste product of purines. We make purine. Our cells, every cell in our body makes purines. Our bags make purines. The waste product of purine is sort of uric acid. And so we secrete that uric acid by urine mostly. And so when the uric acid gets high, the concentration gets high, it comes out of solution, it precipitates and causes gout, right? But so that is known to be pro-inflammatory. But there's this study from 2006 from 2020 here, that showed that even concentrations of uric acid, within the soluble range, concentrations that you don't have gout, but you have high levels of uric acid, can still promote atherosclerosis via activation of sort of inflammatory pathways. And so that was sort of like kind of supporting like sort of these findings that we had. But then sort of the questions that we had, and we thought the reason we thought that this was interesting because this suggested that the gut microbiota actually was modulating the levels of these purines in the circulation and the levels of uric acid in the circulation. So we're interested in understanding how. And so then we're interested in understanding what are the consequences of sort of microbial modulation of these purines, to the host. So this is sort of to bring everybody to the same page for the uric acid. As I said, is the end product, is the waste product of sort of purine metabolism, and we excrete about 70%, in the kidney, but actually 30% of it gets dumped into the intestine. So and there are non-transporters in the intestine cell wall that are known to transport purines, in one or both directions. So this is what happened in human and primate. So mice, well, actually, sort of during evolution, humans lost an enzyme called uricase. So, so this is hominoids. So in hominoid evolution, this uricase gene accumulated several SNPs that basically render this enzyme inactive. So all other animals sort of like have this uricase. So then their waste product is not uric acid. They make some uric acid, but they also make allantoin, which is way more soluble than uric acid and it's easier to excrete. But again, both mice and humans excrete, sort of 70% of uric acid goes in the kidney and 30% in GI. Just that the main difference is that the levels for other animals is quite lower compared to humans. In humans, you can have, up to 7 milligrams. You rarely see more than 2 milligrams in mice. Okay, I think I told you. So one trick that we use with very limited success is, and the reason I'm showing this because I'm going to have data with this, is that we can feed the mice oxalic acid to try to humanize sort of the this part, and sort of like and inhibit this uricase enzyme, to sort of bring up uric acid. But like it doesn't have a huge effect, but this is something that others have tested and we did as well. Okay, so this is what I need to do. So the experiment that we set up is the following. We took germ-free mice, ApoE mice, and so this sort of like they develop atherosclerosis, or conventional mice ApoE that also develop atherosclerosis with and without oxalic acid. So what I'm showing you here is sort of cecal content data. So when we look at the gut of these mice and you look at production of purines, you see that germ-free mice have very little levels of purine, and that makes sense because they don't have bacteria. So conventional mice have bacteria that need to make DNA, RNA, and so on, and they have sort of higher levels of these compounds. Same thing with hypoxanthine. Now, when we look at the excretion products of purine metabolism, like uric acid and allantoin, we see the exact opposite, where germ-free mice have higher levels of these waste products, and this is already in cecal. Now, if we look at plasma, so if we look at the blood of these mice, so we see it's sort of a similar pattern where germ-free mice have higher levels of uric acid compared to conventional mice. The other sort of point that I want to show here is that when we do this oxalic acid treatment in conventional mice, so regular raised mice or with with bugs, we don't see any effects. But oxalic acid actually works to increase uric acid in germ-free mice. And that is useful because we can now test how increasing uric acid affects cardiovascular disease. And so what we see is sort of that, when we feed oxalic acid or oxonate, we don't see any difference in conventional mice, but in germ-free mice, we see an increase, increased burden of atherosclerosis. So suggesting that uric acid will exacerbate the development of atherosclerosis, which is sort of consistent with what others, sort of in that previous study that I show you. I show you that. So everything I told you is in mice. So we look at humans, and so this is a collaboration with Fredrik Bajet in Sweden. And so in humans, so we can now sort of take the aorta and look at the section. So what they use in the clinic is one of the things that happen during the progression of atherosclerosis is the smooth muscle cells get confused and they start thinking that they are bone cells, and they start accumulating calcium. And so a useful metric for in the clinic to look at atherosclerosis is, they do like kind of an X-ray, and they look at sort of calcification of the arteries. It's not X-ray, I think it's MRI that they say. But this sort of looking for these levels of coronary artery calcification is sort of a marker for subclinical atherosclerosis. So we look at sort of whether there were any correlations between serum levels of uric acid and and levels of calcium in the arteries, and so we see this positive correlation there. And we also see sort of several bacteria. So this is we have for these 1,000 people, we also have microbiome data, and we see sort of this negative correlation between several Clostridium species, and keep that in mind, please. Clostridium species and sort of on the levels of uric acid. So sort of negative correlation. Okay, so I told you so far that there are positive associations between purine metabolites and atherosclerosis, and that the gut microbiome regulate purine levels in the gut and uric acid in the circulation. So that's sort of the question that we had was, how? And so we came up with, very simple hypothesis that maybe this bacteria, there are bacteria in the gut that can, catabolize purine, decrease the purine burden to the host, and in that way influence systemic levels of uric acid. That sort of sounded like a reasonable idea, not particularly creative, but sort of like fine. But except that when you look at the literature, there's no evidence of bacteria degrading purines anaerobically. So sort of like this is something that has not been described to happen anaerobically. I mean, there's some papers from or not here, absolutely not in the gut. There are papers, from many years ago, with some soil bacteria suggesting that maybe could happen in some biochemistry done, but definitely sort of not gut isolates, sort of like showing that this can be done at all. Okay. So then sort of what we set up was to, that we tried to do some, isolation studies. And so we took advantage of the fact that uric acid is insoluble. So, and so this is if you put, media without a carbon source and uric acid, and then you inoculate with a sort of fecal slurry, a few days later, you see this turbidity and the uric acid is gone. So just that maybe sort of there's someone using the uric acid after doing serial transfer, you can strip plates and get colonies. And so these plates are tricky, and I'm going to describe them because I'm going to show them a lot. So these plates have saturated levels of uric acid. So the plates look opaque. So now when you have a colony that goes there that degrades uric acid, it creates these halos, so that, sort of showing that, the uric acid that is there is gone. And so the plates do not look opaque anymore. So by doing this approach, we isolated a bunch of organisms that actually, they live next door. We have them all in our collections. They are nothing weird. I mean, it's sort of like the common bacteria that have these capabilities. And we can sort of show that, these organisms, by HPLC show that, the uric acid is gone, and that they actually, and in some cases, make acetate from uric acid. And they actually can grow on it. They can use it as both carbon and nitrogen source. Okay, so we thought, okay, so we're isolating all these bacteria. We say, why we keep isolating? We have a lot of bacteria in the lab. Let's just screen what we have. So we started screening bacteria from different phyla that we have in the gut. Yes, and we have in the lab. Here is sort of how they grow with glucose, and everybody's happy. This media. Now we make the same media but without glucose, and instead with that uric acid. And the plates look opaque. And then you see that there's nothing happening here because these guys cannot use uric acid. But then you have these bacterium E. voltii, or C. difficile that are really good at growing. They create these halos, and they are really sort of showing us that they're using the uric acid. Sorry. So having these sort of having these strains was actually quite important for us, because sort of now we can go back to our germ-free mouse model and assess the causality of these strains. Can this strain modulate levels of uric acid in the host? So this is exactly what we did. So we took germ-free mice, and then we kept them germ-free, colonized them with a core community. So these are organisms that I show you that cannot degrade uric acid or purines, and then the same core community plus some purine degraders. What I'm showing you here is sort of the abundance of this bacteria, sort of this core or the purine degraders, in the gut of these mice. And you see that sort of when the purine degraders sort of colonize nicely too. if we take poop from these animals and we run it in the plates that I show you, we see that the germ-free, poop from germ-free mouse doesn't degrade uric acid. From the core doesn't degrade uric acid. From the, when we add the purine degraders, now we see, these halos. Now we look at the cecal contents from these mice. See that sort of, germ-free, as I told you before, very little purines. We put the core community and now purines go up. And this makes sense because these bacteria need purines to make DNA and RNA and so on. But now when we put the purine degraders there, now the levels of, hypoxanthine in this case go down. Similar thing with allantoin, germ-free, so the core goes up, purine degraders goes down. So this is cecal content. Now I show you plasma. So again, core, germ-free. So this is the levels for germ-free mice. When we add the core, levels of uric acid are not changed. When we add the purine degraded bacteria, now plasma uric acids go down. And we actually have evidence now that just adding one bacterium, so this is E. coli, an E. coli strain that is really good at it, is sufficient to lower the levels of uric acid in the mice. So now the question is what are the genes, what are the bacterial genes important for this metabolism changes? So we did an RNA-seq of, so in this case is one of our purine degraded Enterocluster boltii, that we grow it either with xylose or with uric acid. And here on this the right, I'm showing the genes that are 25-fold regulated by the presence of uric acid. Most of these genes, most of these genes are encoded by two adjacent operons shown here. So these two operons accounted for 12% of the total reads of the transcriptome. So they are very highly expressed. When you look at this, what these genes encode for, you see genes encoded for, enzymes involving carbon-nitrogen cleavage, transport of, a lot of transport of selenium, which is transported for purines. These carbamate kinase and carbamate transferase are important, important energy generation steps, allow the bacteria to generate ATP. And so now we have a pretty good idea of how this, we think how, we need to test this experimentally, how bacteria likely taking uric acid, making a glycine intermediate, we think, and generating 2 molecules of ATP per molecule of uric acid that they are degraded. So, I'm stretching, I'm having some molecular hallucinations here, from this annotation to this, and there's a lot that, needs to be tested here. But you can imagine these genes being, important for uric acid utilization. And so that's what I think the take-home message. So, so these are, we did this RNA-seq in C. boltia or E. boltia, whatever you want to call it. And so these genes, we can find it in other organisms. And as I told you, so there was an E. coli strain that, was able to degrade uric acid, and actually it had this gene. And this is great news because we can easily make mutants in E. coli. So now we can delete these genes and test whether, these genes that we see upregulated are important for uric acid utilization. And actually, this is what we see. This is E. coli without carbon and nitrogen. Obviously, there's no growth. Ammonia, it helps a little bit. We add glucose, and they're happily there. Now, it grows well with uric acid, and this is another, purine adenine. And when we knock out the genes, that grows. It keeps growing fine on glucose, and on glucose, but we completely obliterate growth on these purines. So, suggesting that these genes are actually necessary for growth on purines. We can find these genes in a bunch of different bacteria, Proteobacteria and Firmicutes, and in the arrangements in the chromosomes are quite different, but they are all there. And so I'm running out of time. Yeah, okay, 2 more minutes. So the last experiment that I'm going to show you here is just evidence that these genes are necessary for lower uric acid in the host. So we again, so we went back to germ-free mice, either colonized with the core community I described before, or the wild type strain of E. coli that sort of degrades uric acid, or the mutant that cannot degrade uric acid. And we ask, do those genes are important for uric acid, sort of for lowering uric acid in the host? And what we see is sort of when we add the wild type strain, we see a decrease in uric acid level, and that decrease doesn't happen when we add the mutant strain of E. coli, suggesting that these genes are necessary for lowering uric acid in the host. And so I feel like the most important comparison is here, where we have the core and the E. coli strain, wild type and mutant. Okay, so just sort of to sum up, I found I showed you some positive associations between purine metabolites and atherosclerosis. Show you that the gut microbiome regulates purine levels and in the gut, and uric acid levels in the circulation. I show you that anaerobic uric acid degradation, sort of this pathway for uric acid degradation is detected in Proteobacteria, Firmicutes, and Fusobacteria. So 3 different phyla in the lab, and so we have evidence now showing that the impact purine homeostasis locally, both in the gut and systemically, so levels of uric acid. So we think that these genes sort of could serve as marker of purine catabolism in the gut. And we think that sort of the importance of these sort of findings are beyond sort of cardiovascular disease, where we think that uric acid is important, but for things like gout, this sort of could be sort of useful to reduce level of uric acid in patients that have gout. We also think that this might be useful for understanding gut ecology, as uric acid is both a carbon and nitrogen source. And with that, I will thank you for being here for the invitation, and people in the lab that did the work and our funding source.
Okay, maybe we have time for one quick question, ideally from a trainee or a student, but if not, everyone, it's open to. Oh, yeah.
So the purine metabolizers that may be associated with uric acid production in atherosclerosis, do those tend to be modulated by diet, so that one could sort of loop in the fact that if you eat a more vegetarian diet, you're less likely to have atherosclerosis, and is that mediated through these? Have you thought about that?
There are studies out there, and that's actually, and we've seen it with mice, in humans, when we feed inulin and levels of uric acid go down.
Mm-hmm.
They do this, like, they don't change anything in the diet, but they supplement inulin, and uric acid goes down.
Mm-hmm.
So it doesn't necessarily, directly prove what you're asking.
Yeah.
And we actually see similar things with transplanted mice in different diets. Now we went back to our old data sets where we have all sorts of dietary interventions, and we see uric acid changing in situations where they're not, there's no changes in the purine content in the diet. Just the type of fiber is changing.
Yeah.
Like inulin versus pectin.
Yeah. Very cool.
Other questions? If you're curious about Federico's work, he's gonna be here for a little bit, so you can approach him